This video is Membrane Transport 1. This comes from Chapter 12 Material, in particular the section on Principles of Transmembrane Transport. And as you can see, some of the things we're going to be looking at in detail are transporter proteins and channel proteins. This is follow-up from some of our Chapter 11 material when these types of membrane proteins were first introduced. So I want to call out a couple of the key concepts here, just so you know where this is going. Basically, again, our focus is going to be membrane transport proteins and comparing simple diffusion with facilitated transport. We're going to talk also about permeability and membrane potential. And one reason why all of these concepts are really important is that they're key for cells to live and survive. In particular, for membrane potential, this plays an essential role in neuronal cell signaling, and we'll hear more about that in the future. So here we're looking at some membrane transport proteins, and a reminder about them. So membrane transport proteins, in particular, we're seeing here in this green blobs in the cell membrane. And as you know from our previous material, one way that we can study and understand a little more about how proteins work is using liposomes. And we could compare and contrast a liposome, which in this case is protein-free, versus a cell membrane. And you can see from this picture that due to the membrane transport proteins, solutes can move in or in and out of the cell, whereas for this protein-free liposome, nothing is moving in and out. So membrane transport proteins, these will span the lipid bilayer, and they provide passage across the membrane for select substances. Each type of membrane transport protein could have a different substance that can allow across the membrane. Typically, these are pretty limited. For a given membrane protein, it would allow, say, one type of molecule, an ion or a solute, to move across it, or they could be based on charge, so they may have some more flexibility. We'll give some more specific examples soon. So examples of membrane transport proteins are channel proteins and protein transporters. Both of these are selective. And what we're going to focus on in this chapter regarding these membrane proteins is the movement of inorganic ions and small solutes and water moving across the membrane. A couple other things to mention to you is that you have learned about membrane proteins, in particular in ATP production in the mitochondria, and so these membrane transport proteins are absolutely crucial for allowing those processes to occur. So let's start off by talking about some types of molecular movement. The two that are listed here are simple diffusion and facilitated transport. As you can see, simple diffusion is the process by which molecules and small particles move by random, thermally driven motion. You've certainly heard about this in other classes before, such as introductory biology. And so as a reminder, some of the key features here is that, are that simple diffusion is a passive process. It's spontaneous, and therefore there's no energy required. It's also very slow, and it is not targeted. In other words, this is random movement of whatever molecule we're discussing. Second type of molecular movement here is the facilitated transport process, and this is when the passage of molecules across cell membranes through specialized membrane transport proteins. This is very specific as far as which molecules can move, and it is much faster. This process, depending on the particular membrane protein, may be active, meaning it requires ATP, or it can be passive. Again, it depends on the type of membrane protein. And a couple examples here for when I say membrane transport proteins, what I mean are channel proteins and protein transporters. So something that's going to determine whether a membrane transport protein is required or not is membrane permeability. And so we're going to compare membrane permeability with this figure across several different types of molecules. They're categorized, as you can see, into 4 different categories over here. So basically, we're comparing the membrane permeability, or the likelihood for each of these groups of molecules or ions to cross a membrane. And in this case, they're talking about it as an artificial lipid bilayer, just so we could compare this here. So what exactly is membrane permeability? Membrane permeability, this is the likelihood that a molecule or ion can cross a membrane without a protein transporter. So basically, what's the likelihood that a particular molecule will diffuse across the membrane? And so this is a given factor. This is a known factor for certain categories of molecules, and basically this is determined by size of the molecule and solubility of the molecule. And you can see that over here on the figure that they break it down into small versus larger molecules, for example, and then the solubility part has to do with how it can interact with water. And so you can see up here at the top that we have nonpolar molecules, we have uncharged polar molecules, and then we have larger uncharged polar molecules, and then down here the charged ones. So you should memorize which of these categories and examples within each of these categories can move across the membrane at high permeability, and that basically is the top group here, these small nonpolar molecules. They can easily cross the membrane, and that's indicated by the width of this arrow here. Some things to note in particular are that oxygen and carbon dioxide are on this list. They're small and nonpolar, and they both readily dissolve in lipid bilayers. That means that those gases can be exchanged quite easily across the membrane, which is obviously very important for organisms in their respiration. Then this next group here that has the next level of permeability, so they're not as permeable as the small nonpolar molecules, but they are pretty permeable. These are small uncharged polar molecules such as water, ethanol, and glycerol. And basically, these molecules are going to have some uneven charges, again, because they're polar. So they'll have partial positive, partial negative. So compared to the nonpolar molecules, these will get stuck as they try to cross the membrane, and they don't move as quickly. They can cross, but not as quickly as the small nonpolar molecules. Then next on our list is larger uncharged polar molecules, things like amino acids, glucose, nucleotides, and nucleosides. And so you can see here that the arrow is getting thinner, and a lot of these molecules are getting turned away. And basically these sorts of molecules are typically going to require a transporter. Namely a protein transporter. And later in the course, we'll hear a little bit about glucose. Obviously, that is a common source of energy for cells. Last but not least, this is kind of surprising, ions are actually have the lowest permeability, and so they're almost always turned away from the membrane, and this is because they have a full charge on them, whether it's a full positive or a full negative charge. And basically what that means is that these ions are highly attracted to water, which is going to be, say, on this side of the membrane. And so that inhibits them from crossing into the hydrophobic portion of the membrane. So they get kind of stuck with hydration shells, different water molecules, kind of coating them. And so it's just really unlikely that they're going to be able to move across the membrane. Again, these would require some sort of membrane protein, and a lot of times these would be channel proteins. So highly attracted to water, this inhibits crossing the hydrophobic region of the membrane. Again, these are going to require channel proteins or protein transporters. For example, something like the sodium-potassium pump, which you would have heard about in introductory biology, that is a protein transporter. So next topic is to talk about membrane potential. So we're going to continue to talk about ions and how they move across a membrane. And so basically, membrane potential, that's defined as the voltage difference across a membrane due to electrical imbalances. And I want you guys to notice that when I talk about electrical imbalances or anything that's electrical with a cell, I'm talking about ions, so full positive or full negative charge. So all cells have a membrane potential, and basically a particular cell type is going to have a consistent homeostatic state for that. When we talk about neurons in particular, we'll talk about a resting membrane potential, and this is relative to how a charge will be transmitted across that cell. But for now, notice that this resting membrane potential is a particular ballpark. Depends again on the type of animal cell, what tissue, what type of animal, but it's typically going to be in the range of -20 to -200 millivolts, which that is the abbreviation here. And basically, anytime we have this negative charge for a resting membrane potential, this means that the interior of the cell is more negatively charged than the outside or exterior. And this is going to compare concentrations of ions that are inside the cell versus outside the cell. Basically, how this happens is not accidental. This is due to membrane transport proteins, and they'll maintain this charge difference. So they're responsible for the creation of a resting membrane potential. And they also are responsible for maintaining the charge difference once it gets set up. So to illustrate that, here's an image where we've got the cell membrane here, and then we've got outside the cell, extracellular area, and then inside the cell. And you can see that there are a lot of different molecules that are color-coded here. Some have a full charge, so are ions, and there are some other things listed like proteins here, and it shows that the proteins have a negative charge. So I want you guys to look at that image and answer the two questions here. So just counting up the number of each of the ion or molecule. So what's the most abundant ion outside the cell, and then what's the most abundant ion inside the cell. So pause the video right now to sort that out. Okay, so if we count up on the outside of the cell, there's lots of these blue ions, and so basically those are sodium ions. That's the most abundant ion outside the cell, and this is typical. For cells, the sodium ions would be at high levels outside. You'll also notice that there's a decent number of chloride ions. And this balances out some of the charge difference here. But one thing to keep track of is that, again, sodium is most abundant outside the cell. Obviously, that has a positive charge. That's going to be responsible for maintaining this positive charge difference on the outside of the cell. And then likewise, if we check out the most abundant ion inside the cell, you'll notice that the most common is potassium. Which is a little surprising at first because we see that there's this negative charge inside, but notice that there are a lot of other negatively charged ions and other molecules like proteins that are going to contribute to an overall net negative charge here. So potassium, however, due to a large amount of negatively charged ions plus nucleic acids, so DNA, proteins, and other metabolites. Overall, there's a net negative charge inside the cell. And so part of this is comparing that there's a positive charge over here, which is pretty easy to see, but that we have this a variety of different ways that we have some negative values. So this side is more negative than this side. And the reason I'm telling you about the sodium and potassium is because we'll eventually look at some of the ways that these two key ions are transported, and they're responsible for allowing neurons to fire, allowing our heart cells to work, and so on.